There are three main components of the cell cycle: interphase, mitosis, and cytokinesis. Interphase accounts for 90% of the cell cycle. During interphase, the cell grows and copies its chromosomes in preparation for cell division. Interphase can be divided into three subphases: the G1 phase, the S phase, and the G2 phase. Although during all three subphases, the cell grows by producing proteins and cytoplasmic organelles, chromosomes are duplicated only during the S phase. The mitotic phase, or M phase, is much shorter than interphase. The M phase includes mitosis and cytokinesis. Mitosis is the division of one nucleus into two genetically identical nuclei. The cell starts to divide with mitosis, and cytokinesis finishes off the division. After the cell cycle is complete, there are two cells genetically identical to the parent cell.
Mitosis is important because each new daughter cell receives the same number of chromosomes as the mother cell, and the daughter cells are genetically identical to each other and their parent cells. The speed of mitosis is also beneficial for the cell.
The phases of mitosis include prophase, metaphase, anaphase, and telophase. During prophase, each duplicated chromosome from interphase appears as two identical sister chromosomes joined at their centromeres. The mitotic spindle begins to form. The spindle is composed of the centrosomes and the microtubules that extend from them. Metaphase is the longest phase of mitosis, generally lasting 20 minutes. Centrosomes are at the opposite poles of the cell, and the chromosomes line up in the middle of the cell on the metaphase plate, which is an imaginary plane halfway between the spindles of the two poles. Kinetochores of the sister chromatids are attached to the kinetochore microtubules coming from opposite poles. Anaphase is the shortest phase of mitosis. Anaphase starts when the sister chromatids of each pair separate and become their own chromosome. These new daughter chromosomes begin moving towards opposite ends of the cell as the kinetochore microtubules shorten. The cell elongates, and by the end of anaphase, the two ends of the cell have identical and complete sets of chromosomes. In telophase, a nuclear membrane reforms around the chromosomes at each end of the cell. At this point, the chromosomes become less condensed and mitosis is complete. Cytokinesis is the division of the cytoplasm. In an animal cell, a cleavage furrow pinches the cell in two, while in a plant cell, a cell plate carries out the same function.
The primary result of mitosis is the division of the parent cell's genome into two daughter cells. The daughter cells consist of 46 chromosomes each, similar to their parent cells. Their composition of chromosomes are also identical, which can result in cloning.
The biggest importance of meiosis is that it allows for sexual reproduction, as each parent cell gives a haploid number of chromosomes. Genetic variation results in meiosis due to crossing over of non-sister chromatids, independent assortment, and random fertilization. Genetic variation is important because it drives evolution.
During meiosis 1, homologous chromosomes separate, while the sister chromatids separate during meiosis 2.
In prophase 1 of meiosis, chromosomes condense and homologs loosely pair along their new lengths, aligned gene by gene. Crossing over, the exchange of segments of DNA by non-sister chromatids occurs. Replicated homologous chromosomes have exchanged segments. Each homologous pair has one or more chiasmata, points where crossing over has occurred, and the homologs are still associated. Centrosome movement, spindle formation, and nuclear envelope breakdown occur as in mitosis. Metaphase, anaphase, and telophase of meiosis 1 are carried out similar to the phases of mitosis. In metaphase 1, chromosomes line up by homologous pairs. In anaphase 1, homologous chromosomes separate, and in telophase 1, two haploid cells form while each chromosome still consists of two chromatids.
During prophase, a spindle apparatus is formed. Chromosomes move toward the metaphase 2 plate. In metaphase, the chromosomes are positioned on the metaphase plate. Kinetochores of the sister chromatids are attached to microtubules extending from opposite poles. Anaphase is the breakdown of proteins which hold the sister chromatids together, while the centromere allows the chromatids to separate. The chromatids move toward opposite poles individually. In telophase, meiotic division of one parent cell produces four daughter cells, each with a haploid set of chromosomes. Each of the four daughter cells is genetically distinct from the other daughter cells and from the parent cell. Nuclei also form during telophase.
Meiosis results in four daughter cells in which their chromosome number is reduced by half, and their composition is genetically different from each other.
DNA replication occurs in interphase for both mitosis and meiosis. Mitosis has one division while meiosis has two. Synapsis of homologous chromosomes does not occur in mitosis but does occur in meiosis. Mitosis has two diploid daughter cells genetically identical to parent, while meiosis has four haploid genetically different from parent and each other.